Welcome to the Deep Dive, where we break down a complex stack of research into the essential knowledge you can use. Today, we are taking a massive leap.
A huge leap.
We're going into the molecular blueprint of life itself, the genome, but
Mhm.
we're moving beyond that flat, linear code.
Right.
We're exploring how scientists are finally starting to see the genome in 3 dimensions in its actual functional shape, and how understanding that architecture is, well, it's helping us crack the code of aging.
It really is the highest resolution look we've ever had at biology. We're talking about combining these incredible new profiling methods, how we measure everything, with real breakthroughs in metabolic and epigenetic control.
So it's not just bulk data anymore.
No, not at all. This shift to single-cell, multi-layered information is fundamentally changing how we approach health and longevity.
And as we were digging through all the source material for this, we found a really fascinating, almost bizarre coincidence. It just perfectly sums up this whole moment. Yeah, we found 2 completely separate cutting-edge tools that are, for some reason, named after the same animal. The Coati.
The Coati, you mean that small mammal from Central and South America, the one that looks a bit like a raccoon.
That's the one.
Mhm.
So on the one side, in genomics, you have this new high-resolution method called Hi-Coatis.
Okay.
But then, at the exact same time, in the world of computation, engineers are using what they call the Coati Optimization Algorithm, or COA, to solve these super complex design problems.
That's incredible. So it's a perfect metaphor.
It's perfect. It just shows that this massive push for higher resolution in biology is completely inseparable from the optimized computational power you need to interpret it all.
They go hand in hand.
Okay, so let's unpack this genomic revolution. Let's start with that 3D structure. For so long, we've just thought of DNA as a long string.
The instruction manual.
Exactly. But now we know structure is destiny.
It absolutely is.
Yeah.
That long string of DNA in a chromosome isn't just, a pile of spaghetti in the nucleus. It's this highly organized, functional, hierarchical architecture.
What are those levels of organization?
Well, at the biggest scale, you've got chromatin territories and AB compartments. They basically divide the DNA into active zones and repressed zones. But the real action, the functional units of regulation, are these things called topologically associating domains.
TADEs.
TADEs, yeah.
So if I'm getting this right, TADEs are kind of like neighborhood boundaries. Everything inside the boundary can interact, but it can't really talk to the stuff outside.
That's a great analogy. TADs create these functional units where a gene and its regulatory elements, like an enhancer, can find each other.
And they do that by physically touching.
They do. The looping action is the fundamental principle here. That physical loop brings a distant enhancer right up next to its target gene to turn it on or off. And within those TADs, you have even smaller structures, like subTADs and chromatin loops, that are crucial for things like cell differentiation.
So what happens when a loop fails or one of those boundaries just breaks down? Is that a subtle glitch or a catastrophe?
Oh, it's often a catastrophic miscommunication. The TAD boundaries act as insulators. They block the spread of histone modifications, those chemical tags on the DNA.
They keep things tidy.
They do.
Right.
And if you disrupt a key anchor for that structure, like a CTCF binding site, you immediately change the local architecture. You might accidentally connect a really powerful growth enhancer to a gene that's supposed to be quiet.
And that leads to big problems. This all sounds incredibly hard to measure. I mean, how do you actually see DNA folding?
That's where a family of methods called chromosome conformation capture comes in. We usually just call them the Hi-C methods.
Okay.
And what they do is measure the frequency with which 2 DNA segments that are far apart on the linear map are actually close together in 3D space.
And our sources pointed out how fast these methods are getting better.
They're evolving incredibly quickly. We've gone from standard Hi-C to really powerful variants like Hi-P and Capture-C. The sources mentioned Hi-C 3.0, which shows this dramatic improvement in quality. Yeah. It got up to an 80.7% cis proportion.
Okay, for everyone listening, why is that number 80% cis so important? What does that tell you?
Because cis means interactions happening within the same chromosome. You expect that. Chromosomes kind of stay in their own territories.
Right.
If you see a lot of trans contacts, meaning between different chromosomes, that's often just noise, random DNA junk. So a high cis number means you have a much cleaner, higher resolution map of the functional folding we're talking about.
And now there are even more specialized tools.
Yes, like the Capture-C family. They use these synthetic oligonucleotides to basically fish out and enrich for interactions you're specifically interested in. And variants like Ali Capture-C let us do this with really, really small numbers of cells.
Which brings us right to single-cell analysis. That has to be the next step, because if structure is destiny for a cell, you need to know what that structure is in every single cell, not just the average.
Precisely. Classical bulk methods give you the average across millions of cells. In a complex tissue like the brain or a tumor, that average just hides all the important differences. It masks the cell-to-cell heterogeneity.
It misses the rare cells that might be causing the problem.
Exactly. Bulk data just doesn't have the resolution, and it needs way too much starting material.
But I imagine single-cell epigenomics it's a technical nightmare. You're working with vanishingly small amounts of DNA.
The challenges are immense. You've got high variability between cells, really low coverage, meaning you only see a tiny fraction of the genome in each cell, and it's expensive. Plus, when you amplify that tiny bit of DNA, you can introduce huge biases.
So let's talk about how they're getting around that. For something like DNA methylation, the classic method is bisulfite conversion, which is pretty harsh. What are the alternatives?
We're seeing a big shift toward affinity enrichment methods. So instead of a harsh chemical treatment, these methods use proteins or antibodies that literally grab onto the methylated DNA segments, things like mita-heuristic or MBD-seq.
What's the trade-off?
The big trade-off is resolution. You don't get that single base view that bisulfite can give you.
So how do you choose which one to use?
It really comes down to CpG density. MBD methods are great for dense clusters of methylation, these things called CpG islands or CGIs. Think of them as finding the cities of methylation.
Okay.
Mita-heuristic, on the other hand, is better for the less dense regions, the sort of suburban sprawl of methylation. It's all about context.
And to get around that amplification bias you mentioned, especially for single cells, we're seeing new tricks like CCGI-seq.
Yes, CCGI-seq is a bisulfite-free method that uses something called multiple displacement amplification, or MDA, to reduce that PCR bias.
Right.
And it works. It gets incredibly high coverage for a single-cell method, over 70% of all CGIs in one study. That's a huge leap in data quality.
So once you have the methylation map, you need to know which parts of the genome are actually open for business, right?
Right. You need to know which parts the machinery can access and read.
And that's where ATAC-Seq comes in.
Absolutely. ATAC-Seq is the go-to technique for mapping open chromatin. It's like getting a floor plan of the cell's library, showing all the unlocked doors. It tells you which genes are active or, just waiting to be transcribed.
And then the dream is to combine all of this, right? The structure, the methylation, the accessibility, the final output, all in one cell.
That's the holy grail, single-cell multi-omics. Quantifying different types of molecules, chromatin, RNA, whatever, all at the same time from the same cell. It gives you a complete holistic view of what that cell is and what it's doing.
The sources mentioned a workflow called MUSIC.
Yes, MUSIC is a great example. It's a workflow that measures multiplex chromatin and RNA interactions, and they've used it in really tough systems, like aging human brain cells. Get the whole story of that cell's life.
Which is absolutely essential for understanding aging, because at its core, aging is driven by this progressive epigenetic dysregulation.
That's the consensus, yeah. Aging involves all these changes. DNA methylation goes haywire. You get abnormal histone modifications. The whole chromatin structure remodels.
And we can measure this with the epigenetic clocks.
Exactly. These are DNA methylation-based biomarkers that can estimate your biological age, which is often very different from your chronological age.
And the functional results of losing this epigenetic control can be devastating.
They really are. In neurodegeneration, for instance, there's a specific loss of a histone mark called H4K12-ACI in the hippocampus.
And what does that do?
It silences the genes responsible for synaptic plasticity, the very genes your brain needs to learn and adapt. When they go quiet, cognitive decline speeds up.
The physical changes in senescent cells, the ones that stop dividing, sound even more dramatic.
Yeah.
You mentioned these cytoplasmic chromatin fragments, CCFs. What on earth are those?
So senescence is a total reorganization of the nucleus. For one, you lose the connection between lamoni B1 and the tightly packed heterochromatin.
Okay.
But the CCFs are even crazier. They are literal fragments of chromatin that pinch off from the nucleus and escape into the cytoplasm.
So you have DNA just floating around in the cell where it's not supposed to be. Why is that so dangerous?
Because your cytoplasm has sensors that are designed to detect foreign DNA, like from a virus. So when these CCFs are floating around, rich with repressive histone marks and DNA damage markers,
The cell thinks it's under attack.
it thinks it's under attack from itself. It activates this innate immune pathway called CGA-STING, which triggers this chronic low-grade inflammation that is a major driver of aging.
So if epigenetic control is the problem, we need a control center to fix it. And the sources point to a really interesting one, the hypothalamus.
It's the ultimate regulatory hub for keeping the body in balance, for homeostasis. And researchers found that mTOR activity, that key nutrient-sensing pathway, really ramps up in the hypothalamus of aging mice. And that contributes to things like obesity.
Which makes it a perfect target. And that brings in a class of drugs that most people know for weight loss GLP-1 receptor agonists.
Right, things like exenatide. But the key insight from the research is that they're not just metabolic drugs. They seem to be molecular age-counteracting agents.
How so?
In aged mice, these GLP-1RAs produced multi-omic signatures that looked remarkably similar to what you see with rapamycin.
Which is the gold standard mTOR inhibitor for anti-aging research.
Exactly. And the effects were almost entirely dependent on the GLP-1 receptor in the hypothalamus. It suggests you can target this brain-body axis to fight systemic aging.
And this is starting to translate into clinical use, right? For things like hypothalamic obesity.
It is, though the results are a bit variable. One case series we saw was really promising. 8 out of 9 patients with HO saw substantial weight loss on GLP-1RAs. It shows the potential of hitting that central switch, even if we need to refine the protocols.
But it's not just drugs. The environment itself can act on the 3D genome.
That was one of the most exciting findings. A study using promoter capture Hi-C showed that just putting old mice in an enriched environment with more stimulation, more to do, it could partially revert the age-related changes to their 3D chromatin structure in the hippocampus.
So the genome is dynamic, it can be rejuvenated.
Yes, and that rejuvenation extends even to our vasculature, our blood vessels.
How so?
High-resolution 3D imaging shows a hallmark of aging is the loss of blood vessels and parasites in most organs.
Yeah.
The kidney, spleen, brain. But here's the kicker. In tissues that are constantly remodeling, like the gut and the skin, the vasculature is preserved.
Which suggests their regenerative ability is tied to maintaining that blood supply.
It seems to be yes.
Okay, so all of this research generates just a staggering amount of data, which brings us full circle back to our mascot, the Coati. Let's define the first one, Hi-Coatis.
Hi-Coatis. It stands for high-throughput capture of actively transcribed region interacting sequences. It's an ingenious method.
What makes it so clever?
It combines the detection of active transcription with 3D chromatin interaction studies all in one go. And it does it by capturing signals from all 3 major RNA polymerases.
So it's incredibly efficient.
Massively efficient. It doesn't need specific antibodies or probes, and it manages to capture over 93% of all expressed genes in a single assay. It's a huge boost for understanding the working architecture of the genome.
And all that beautiful high-res data needs equally advanced tools to make sense of it. That brings us to our second Coati, MACOA.
The multi-strategy adaptive Coati Optimization Algorithm. This is a meta-heuristic algorithm from the engineering world, and it's built to solve a problem that's common to all big data.
Which is what's
getting stuck. Think of optimization as trying to find the lowest point, the best possible solution in a huge, dark, mountainous landscape. The standard algorithm often finds a little ditch.
a local optimum.
Exactly.
Yeah.
It thinks it's found the best answer when a much deeper valley exists just over the next ridge. The original Coati algorithm had this problem.
So how does MACOA get around that? How does it find the true global best?
It uses 3 main strategies. First, it uses something called levee flights, these long, random jumps, to explore the whole landscape from the start. Second, a nonlinear inertia weight helps it fine-tune its global search.
And the third?
The third is my favorite. It's called the Coati Vigilante mechanism. It basically forces the algorithm to periodically take these big exploratory steps to make sure it's not trapped in a local optimum.
It's forced to be adventurous.
Right. And the result is a massive improvement in optimization speed and performance. It's exactly the kind of smarts we need to analyze these huge, sparse, Hi-C data maps.
It's the perfect illustration of that partnership. You generate this incredibly complex 3D data, and then you need sophisticated AI, like deep learning models, to enhance its resolution and actually interpret it.
That synthesis is the whole revolution. It's these multi-layered, single-cell 3D perspectives on life, and they're all driven by technology that's often borrowed directly from computer science.
So we've seen the techniques that map life in 3D, the specific changes that drive aging, and even some molecular levers like GLP-1RAs that can counteract it. We are gaining a lot of control.
We are. And that control leads us to a really urgent question for the future. The power to reverse aging at the molecular level through really aggressive epigenetic therapies like HDAC inhibition or partial reprogramming, That power is getting closer.
But what are the risks that come with that kind of fundamental control over the genome?
The main risk that keeps coming up is malignant transformation, tumorigenesis. If you apply a broad, system-wide epigenetic reversal, you run the very real risk of waking up powerful oncogenes that should be silent.
Or silencing the tumor suppressors that are supposed to be protecting you.
Precisely. Which leaves us with the critical question to think about. As we gain this incredible power to rejuvenate the genome, how can we ensure precise tissue-specific control? How do we maximize the benefits of reversal while completely avoiding the fatal outcome of malignant transformation?